Hello, my name is Frank Baganz and the title of my talk is Characterization and Scale Translation of a Parallel Microbioreactor System for Cell Culture Process Development. This work is part of an engineering doctorate project between UCL Biochemical Engineering and Applicon Biotechnology. The aim of the project is to establish the micromatrix as screening tool and scale-down model for cell culture applications. In this talk, I will present the work on the engineering characterization of the micromatrix, focusing on mixing time and KLA. Then I will demonstrate how matched mixing time in combination with a similar CO2 addition profile can be used to scale down a fed batch process using a FHO cell line producing an IgG4 antibody from a stirred tank benchtop scale reactor to the shaken micromatrix system. Subsequently, the high throughput of the microbioreactor will be used to perform a DOE-assisted optimization of the feeding strategy. Lastly, the optimized fed batch protocol will be translated to the benchtop scale. In a typical process development timeline, clone screening and selection is usually done in these 96 well plates. Promising clones are then transferred to shake flask where the medium composition can be optimized. The process is then transferred to a benchtop scale stirred tank reactor where process parameters such as pH and dissolved oxygen can be actively controlled. Finally, the translation to pilot and production scale follows. Both throughput and the amount of information that can be obtained from a run are functions of the scale. The smaller the scale, the more experiments can be run in parallel, but also less information is obtained. At the larger scale, financial and space constraints restrict the number of parallel experiments. However, these experiments allow for better insight at conditions closer to the actual production process. Microbioreactors, like the micromatrix shown here, try to shake up this inverse relationship by offering a fully controlled culture environment at small scale that allows for a high degree of parallelization. The micromatrix is designed around a 24 deep square well microtiter plate, as shown here. On the right, you can see a close-up of one of the 24 wells. Each of the wells has a filtered gas inlet for the addition of up to 4 gases, a gas outlet and a liquid line which allows for feed addition but also pH control. On the bottom of the well are sensors for the measurement of pH, DO, and temperature. The microtiter plate sits inside the micromatrix on an inbuilt orbital shaker. An engineering characterization of the micromatrix was conducted in order to provide the basis for the scale down of an existing fed batch process in a 5-liter stirred tank reactor to the micromatrix in the shaking 24 standard round well format. This photo shows the setup of the mixing time experiments using a mock-up well of the same dimensions as the micromatrix cassette wells. The well was fixed onto an orbital shaker. The color change of a dual indicator system for mixing time solution was used to determine the mixing time inside the well under dynamic conditions. Pictures were taken using a high-speed camera mounted on the shaking platform, shown here. The mixing time was determined by analyzing the pictures and visually observing the color change. Mixing time tests were performed for different values of volume and steering speed. The mixing time as function of the rotational speed as shown in the figure here, was found to range between 1 and 42 seconds. An increased working volume generally led to longer mixing times, whereas increased shaking speed shortened mixing times. This is an expected result, and it also agrees with previous studies where a similar trend was demonstrated, as shown in this table for different miniature bioreactor systems. This photo shows the setup to determine the volumetric oxygen mass transfer coefficient, KLA. Measurements were performed using the static guessing out method. To perform the measurement with a high frequency and with a sufficiently short probe response time, a needle-type oxygen microsensor was used, as shown in this photograph. The probe was inserted through the off-gas filter of the assembled micromatrix cassette. All dissolved oxygen was first removed from the liquid through overlay with nitrogen. The headspace was then flushed before the operating conditions with regard to airflow rate and shaking speed were set and recording commenced. The results are illustrated in these figures, where the KLA is plotted as function of shaking speeds for 3 different airflow rates. The values of the KLA ranged between 2 and 240, depending on the operating conditions used. The mass transfer is directly proportional to the surface area of the liquid. With increasing shaking speeds, the liquid surface gradually deformed and its inclination grew, which led to larger liquid surface area and therefore higher KLA values. The KLA had an inverse relationship to the liquid volume and in turn decreased with increasing working volume. However, there was little effect of changing the airflow rate on the KLA, as you can see from these graphs. There are 4 different criteria that are commonly used for scale translation of cell culture processes. The first is based on matched tip speed. However, this is not applicable for scaling between shaken and stirred systems. The second is based on matched power per unit volume, but this value can only be derived via simulation or elaborate experimental setup for shaken systems. This leaves matched KLA or matched mixing time as potential candidates for scale translation between stir tank reactor and the micromatrix. This table shows the operating conditions for the 3 different cultivation systems used in this study. A KLA value of 1 per hour is often cited as being sufficient to support cell densities of up to 1 × 10⁷ cells per milliliter, and all cultivation platforms fulfill this requirement. Therefore, a matched mixing time of approximately 6 seconds was chosen as criterion for the scale translation from shaken microwell systems to the benchtop scale. This figure shows the viable cell concentration and viability of a GSCHO cell grown in the 5-liter stir tank reactor and the micromatrix system. Also, the viability is very similar. The maximum viable cell concentration is more than double in the micromatrix compared to the 5-liter reactor. In contrast, the IgG4 antibody titer is almost identical. The results suggest that similar mixing conditions alone do not promote similar cell growth. Closer inspection of the carbon dioxide addition profile showed large differences between the stir tank reactor and the micromatrix. This figure shows the CO2 is continuously added via overlay in the micromatrix due to the manual bolus additions of base. By contrast, in the 5-liter stir tank reactor, CO2 addition stopped after 3 days due to lactate formation. Further acidification is counteracted by base addition. When lactate is consumed towards the end of the run, CO2 addition started again as shown in the graph. Such a CO2 addition profile was emulated in the micromatrix by using the automated feeding module instead of manual bolus additions of base. As you can see, for both CO2 conditions, the growth and product profiles are comparable between the 5-liter stir tank reactor and the micromatrix. Furthermore, the maximum viable cell concentration did not show significant differences for these systems. Also, the growth of cells cultivated in the 24 standard round wells was slightly slower. The trend was comparable to the cultivations that used gas blends with 5% CO2. Similarly, the maximum viable cell concentration in the 24 standard round wells was reached at a later stage of the cultivation, but was not significantly different to either the micromatrix or 5-liter stir tank reactor. In addition, the final titer was not significantly different between the scales and CO2 conditions. This indicates a difference in the cell-specific productivity caused by the difference of the CO2 concentrations in the inflowing gas. This table shows a summary of the results from the previous slide, confirming the impact of the CO2 addition profile on growth and product formation in the micromatrix and 5-liter stir tank bioreactor systems. It also shows a high degree of similarity between the two controlled bioreactor systems for all derived parameters. By contrast, some larger differences in growth rate and final titer were found when cells were grown in the 24 standard round well system. In order to ascertain whether the N-linked glycosylation of the IgG4 antibody was scalable, the distribution of key glycoprofiles was compared. As you can see here, the percentage of fucosylated glycans was above 95% at all scales, which was similar to earlier studies of the same cell line and product. The distribution of sialylated antibodies was similar for the controlled systems, but reduced for the 24 standard round well format, as you can see in this graph. Similarly, galactosylation was comparable for all conditions run in the micromatrix and 5-liter stir tank reactor, but considerably reduced for antibodies that were produced in the 24 standard round wells. This was likely caused by a difference in the pH profile, because cultures in the 24 standard round wells were not pH controlled. In summary, the results show that a matched mixing time in conjunction with a similar CO2 addition profile proved to be a suitable scaling criterion to translate a fed batch process from a conventional stir tank bioreactor to the micromatrix system. Having demonstrated the scale translation of a fed batch process, we used the micromatrix to optimize the bolus feeding strategy. A design of experiment approach with a circumscribed central composite design was employed to investigate 3 factors. These were the total feed volume in percentage of the initial working volume, the feed start in days after inoculation, and feed weight in number of bolus additions that the feed volume was divided. All were investigated at 5 different levels. The center point was replicated 6 times. This resulted in 20 runs in total, performed in a single experiment. The feeding regime was optimized for the space-time yield instead of the final titer in order to avoid a bias towards long process run times. The contour plot here shows the optimal space-time yield was captured within the investigated design space. The optimal value of 71 milligrams per day and liter was predicted for a feed start after 5 days and a total feed volume of 64% that was divided into 6 bolus additions. The predicted optimal bolus feeding regime was repeated in the micromatrix for validation and then scaled up to the benchtop bioreactor. A comparison to the original fed batch protocol, shown as historic in gray, was made to assess the success of the optimization. The results show minimal differences of the growth kinetics between the scales for the optimized feeding regime. In comparison to the original feeding strategy, a moderate increase of the maximum viable cell concentration and a prolonged stationary phase were observed. Similarly, the progression of the titer was nearly identical between the scales for the optimized feeding regime and significantly higher compared to the original fed batch regime as shown in this graph. These results are confirmed by the comparison of the derived parameters that were almost identical between the micromatrix and the 5-liter stir tank reactor under optimized conditions, whereas the values for the original feeding regime showed a substantially reduced productivity, as shown in this table. The final titer and space-time yield of the optimized feeding protocol was about 20% higher compared to the original protocol and very close to the predicted optimum. In conclusion, initially, an engineering characterization of mixing time and KLA was performed to gain an understanding of the cultivation environment generated by the system. Mixing and KLA were found to be comparable to other microbioreactor systems and lab-scale bioreactors. Based on the information obtained from the engineering characterization, the mixing time was selected to remain constant between the 5-liter STR micromatrix and 24 standard round well to ensure comparable hydrodynamic conditions. Furthermore, the growth kinetics of the used cell lines were highly dependent on the CO2 addition profile. As a result, the concentration of CO2 in the inflowing gas was also matched between the shaken micromatrix and the 5-liter stir tank reactor, leading to very similar growth and product kinetics as well as glycosylation profiles for both CO2 conditions. The high throughput of the micromatrix was then leveraged to optimize the bolus feeding strategy within the framework of a circumscribed central composite design. A scale-up of the optimized feeding regime demonstrated nearly identical growth and production kinetics between the micromatrix and the benchtop scale reactor and resulted in a 20% increase of the space-time yield compared to the original feeding regime. The results demonstrate the potential of the micromatrix system, a scaled-down model for fed batch cell culture processes and its application for process optimization. All the work presented was carried out by Vincent Wiegmann with support of Dr. Gregorio Rodriguez and Ms. E. Lee in setting up the mixing time experiments. I would also like to thank Applicon Biotechnology for provision of the micromatrix system and financial support of the NHD studentship. I would like to thank Lutka Limited for their help with the glycosylation analysis and Lonza UK for provision of the GSShow cell line. This work was supported by the UK Engineering and Physical Science Research Council as part of an Engineering Doctorate Training Center grant in Bioprocess Engineering Leadership. Finally, thank you for listening to my presentation, and I'm happy to answer any of your questions.